Today we're going to look at DNA structure, and then we're going to transition into replication, and we're going to start the process of protein synthesis just by introducing RNA and its general overall reason for existing. So let's first focus in on DNA structure, and DNA structure is very much what you learned in baby bio. There's not really much more for me to say here. You have what we call nucleotides. So this is a nucleotide. Nucleotide consists of three parts: a phosphate group, a sugar, and in DNA that sugar is deoxyribose, and then a nitrogenous or nitrogen-containing base. So phosphate, sugar, base was how I asked you to remember it in baby bio. Now, the sugar we need to focus in on for just a minute because there's this funny 5' 3' business that we need to talk about here. When we are looking at the sugar, we start counting the carbons in the sugar. This is the first carbon, carbon 1. Carbon 2 is here. This is what we call the 3' or third carbon. Fourth carbon is here. And then this sticking up carbon is the 5th carbon in this sugar. This is called a pentose, okay, because it's got 5 carbons in it. And it's this 5th carbon that we call the 5' carbon. Now, the reason why that's important is remember that the strands of the DNA molecule run what we call anti-parallel, right? Because this strand is running with the 5' end of the sugar sticking up, and then down here at the bottom is where you'd find the 3' end, right? The other strand, 5' end, starts down at this side, and then the strand moves in the opposite direction with its 3' end sticking up. So this sugar is flipped upside down, which means the whole nucleotide is flipped upside down. Okay, so the concept of being anti-parallel we showed you in baby bio, but we didn't really count the 3', 5' carbons, but we need to understand that here because that's going to matter in terms of directions when we're talking about replication and transcription. All right, now remember that the strands of the DNA molecule, basically you take a nucleotide and you attach it to another nucleotide and another nucleotide. This is a chain of nucleotides or a strand on the DNA molecule. The phosphates and sugars on that strand have this repeating segment, and that we call the backbone of the molecule. So on this diagram here, here's the backbone written in red, right along the edge, here and here. And then here you can see that that backbone is consistent or consists of a phosphate and a sugar. The important part of the molecule really is the base, right? Because it's the base, the order of these bases that determines the code on the DNA molecule. And that's going to determine what's on the other side, right? What's on the other strand. Because T has to bond with A and G has to bond with C, right? That's the complementary base pairing rule that you guys learned way back in baby bio, and you can see that here. Here is a T, it's bonded to an A. Here's a G, it's bonded to a C. And everywhere you look on this molecule, you see that configuration, even if it's flipped, where the A is on this side and the T is over here. Same thing here, G is over here, C is over here. But you're always binding A to T and G with C. That's because A and G are what we call purines. And notice the purines have 2 rings of carbons, right? They always bind to a pyrimidine, which are the T and the C. And you can see that depicted here. Notice this longer segment on the A is showing you that that's a purine. It's got 2 rings of carbon, so it's longer. The pyrimidine is shorter, right? Same thing here. The G is longer, the C is shorter. This idea came from Chargaff, that's how you had to put this molecule together, because if you bonded an A with a T or with a G, you'd have a long segment here, and then if T went with C, you'd have a short segment, so you'd have a molecule that kind of had this funny shape where the helix would be bent in like this, right? because of the cross sections, right? If this were A and, or C and G here, and this were, or C and T, sorry, and this were A and G and so on, the molecule would get wide at certain areas and narrow at other areas. And Chargraf said that really didn't seem to happen. The molecule, in order for it to be straight up and down, as you see pictured in this diagram, you had to bind a purine to a pyrimidine. He also noted that the adenine and thymine occurred in about the same ratios as the guanine and cytosine, and he used that to kind of intuit that, in fact, that's the complementary base pairing rule. Now, of course, the last thing that's pictured here is that this ladder-like structure of DNA that you're seeing actually twists, and that's the helix part, right? So it's double because you have two strands that run anti-parallel. The helix is that it twists. All right, now before we go any further, it's really important for you to note that College Board is very specific about what comes next, and there are a ton of enzymes. I always call it a symphony of enzymes involved in any cellular process, really, but especially that's true with replication, with transcription, and translation in the next video as well. So when we're doing these processes, you need to be aware of what DNA polymerase is, ligase, helicase, topoisomerase, and then when we get to transcription in the next video, you'll need to know about RNA polymerase. But polymerases kind of do the same thing, so whether it's RNA polymerase or DNA polymerase, you'll pretty much understand it either way. Now, I will say there are several types of DNA polymerase. There are several types of RNA polymerase. You just need to know in general what a polymerase does. These are the 5 specifically that College Board mentions. So make note of them here, get them down, and as I describe this, even though you'll see other things in the pictures, you don't need to worry about the other enzymes that you see depicted in the pictures. All right, so let's start with replication. First of all, remember, replication is special. The only reason for replication is in preparation for cell division, okay? That's why I'm kind of keeping it separate from the rest of what we do with DNA, right? That's why that's in another video. Because replication is only there to make copies of a DNA molecule so that you have 2 copies, one to go in each of the baby cells that you make in mitosis, for example. Okay. Of course, I just mentioned there's a whole bunch of enzymes involved in this process, but you don't have to worry about what those enzymes are, except for the 5 that I've mentioned. The direction here, you're starting at the 5' carbon, and you're moving in the direction of the 3' carbon. That's what we mean when we say it's a 5' to 3' direction. And notice that means replication doesn't go down the line like this. One enzyme attaches to one strand in one direction and it attaches to another strand, or the enzyme attaches in another direction. And actually, it's a bit more complicated than that. And I'll kind of explain it when we get there, and then I've got another video that I've posted for you to watch as well, where you can see this in real time and you can understand why there's a difference in the way that one strand is copied. So right off the bat, you're going to see this is a little bit more complicated than what you had in baby bio. Because one of the strands gets to be copied continuously. Because really, if an enzyme is here and it's copying this strand, if the enzyme is the information is being fed in correctly in the correct direction, and the strand or the enzyme is able to move this way, the same enzyme can't copy this strand the same way. So this is the leading strand, and then the other side we call the lagging strand because it's got to be copied. It's entering the enzyme backward, basically, and it's going to be pulled out in loops and kind of replicated in fits and starts. And like I said, I'll get into that in a minute. We also call this semi-conservative because remember, each new piece of DNA that we make, if you look at it right here, this is our old original piece of DNA, right? The new DNA is represented in red right here, right? Meaning each copy that we make has a strand of the original piece of DNA and then a strand of new DNA in it. That's what we mean when we say it's semi-conservative. Now, In bacteria, you have one single point of origin. Since you've got these circular chromosomes, there's a single point of origin, and it just goes around, around until the whole thing is copied. It proceeds in both directions. In eukaryotes, what you get are multiple replication bubbles. Several enzymes will clamp down and will start replication in what we call a replication bubble. Also, you have to understand that there is a primer. There's an RNA segment that is attached to start the process, because the enzymes that synthesize DNA cannot attach nucleotides to nothing. They have to have something to attach it to. So that primer, that piece of RNA, is actually attached right here at the beginning. And then, of course, this is going to end with proofreading. There are going to be enzymes that are going to come behind and check and make sure that you have the correct bases in place. Okay, let's start with the synthesis of the leading strand. And for the leading strand, remember, you need helicase and topoisomerase, and then you're going to need a DNA polymerase. Now, this is DNA polymerase 3. Remember, I said just know DNA polymerases in general, right? Okay, so helicase. Let's start with topoisomerase, because topoisomerase is going to attach first, and it's going to break and rejoin the double helix to kind of relieve the torsion force on the molecule, because the molecule has to be untwisted in order and opened up in order for copying to begin. So topoisomerase is going to start to open up the DNA by kind of breaking and rejoining. Really, it's the backbone of the molecule that it's working on. And then along behind that comes helicase, and this is going to start breaking apart the hydrogen bonds between the two strands of the DNA molecule. Now, again, you've got some other proteins here, like the SS BPs, and you've got primase and a sliding clamp and things like that. But for now, that's all you need to worry about, plus DNA polymerase. So like I said, this is the easy strand, right? This is the strand that's actually going in the correct direction. Now, I misspoke on the previous slide. I said that it's synthesized in the 5' to 3'. That means the new strand is made in the 5' to 3' direction. The DNA molecule has to enter in the DNA polymerase molecule that's going to actually add the new nucleotides in. It has to enter from the 3' side. Okay, so this top strand is the leading strand of the molecule. Okay, so the polymerase molecule would come along from here and it would move in this direction. Okay, and that's what you're seeing right here. Up at the front, you see topoisomerase working. You see helicase breaking apart the bonds, and then this begins with a primase molecule, which I mentioned before. That's going to put a primer on the molecule to start the new strand. And then DNA polymerase can attach right here and start sliding along, and as it does, using the complementary base pairing rule, it's just going to add nucleotides into to add on a new strand to this molecule. Okay, now that new strand is synthesized 5' to 3'. Okay, but it's actually sliding along the leading strand from 3' to 5'. Okay, directions get kind of complicated here. Okay, but when we say that the DNA polymerase works from 5' to 3', we mean that the new strand that is built from 5' to 3'. Okay, now let's take a deep breath, because we are now looking at the lagging strand. Okay, now the lagging strand, where the leading strand went in the leading strand entered the DNA polymerase enzyme the correct way, right? On the lagging strand, because the strands are anti-parallel, now what's happening is the enzyme, or the DNA strand that we're trying to copy, is entering the enzyme backward, essentially, or the enzyme complex backward. And so what happens is you have to kind of pull out the strand and re-loop it in correctly. But what that means is that you end up copying little tiny fragments of the DNA molecule at a time on that lagging strand. Okay, so we start the same way. We have topoisomerase, right, releasing the torsion pressure on the molecule. We have helicase breaking apart the hydrogen bonds, just like before. This time now, on this strand, primase is going to create the primer, right? But this time now, you're going to have the molecule, this same enzyme and its sliding clamp attaching and moving the other way. Okay, and it's really, you can't really see very well here why you need multiple fits and starts. And this is where the video that I will show you or ask you to look at after this is a little bit more descriptive. But essentially, when primase comes in, it actually attaches at multiple points downstream, creates a primer, and then from that primer onward, a DNA polymerase molecule can attach and move to create a new small segment of the brand new piece of DNA. Okay, so what you end up with are these little fits and starts, and that's why it's called the lagging strand. In this case, what happens is you end up with what we call an Okazaki fragment. Okay, Okazaki fragments are these little pieces of DNA where you end up with a piece here of one strand, then it stops, and then you get the next piece, and that's the next fragment, and so on, and it goes down the line that way. Now, at the end of this whole process, the leading strand is complete, right? But the lagging strand still has these little primers attached, right? And really, the leading strand has a primer on it as well, and this is going to happen with the leading strand too. There is a molecule that's going to come along and take off another kind of DNA polymerase that takes off the RNA primers and now replaces them with DNA nucleotides, right? And then there's a new molecule, and this is another one that you need to know about. This is ligase, right? And ligase is going to go in and attach the backbones of the Okazaki fragments together. So after the DNA polymerase molecule that finishes up work removes the primers, leaving gaps here, not gaps, really leaving new pieces of DNA, something's got to come in and repair this break in the molecule, and that's what DNA ligase is going to do. Now that's not all, because obviously sometimes mistakes can be made in this process, and when mistakes are made, something has to come along and repair those mistakes. So notice there is another molecule, and usually it's another kind of DNA polymerase that can come in and notice there's some problem, right? Here are some damaged bases. You've got a little bend in the molecule backbone right here. Something's wrong here. And what happens is, or sometimes you'll have even some mismatched bases, like you'll have a G attached to a T or something like that. An enzyme comes in and pulls out the damaged section, and then a DNA polymerase comes back and fills in the gap. And then again, a ligase molecule has to join those gaps together, and now you have repaired damage. All right, now remember, this was a process that we went through in order to do mitosis, right, or meiosis, because you replicate DNA in both of those processes right before both of those processes begin. After replication occurs, typically what's next is to create chromosomes out of them. Remember, the DNA most of the time exists as chromatin. It's all unraveled, uncoiled, and so on. And so what you need is a way of coiling them up. And remember, the reason why we coil up the DNA into chromosomes is to make them easier to move, right? So I thought now would be a good time to remind you of how these things relate to one another. So essentially, after DNA is replicated in preparation for cell division, well, actually, all the time, the DNA gets wrapped around these globular proteins, these globe-shaped proteins, and there are actually 8 of them. 1, 2, 3, 4 here, and then there's 1, 2, 3, and then a 4th one right back here on top. These are called histones. Okay, so there are 8 histones per complex here. And the DNA kind of wraps around those histone proteins. And this DNA wrapped around histones is what we call chromatin. Each histone complex with its associated DNA is referred to as a nucleosome. Okay, but when DNA is in this chromatin form, it's basically DNA that's wrapped around histones. In preparation, though, for cell division, the chromatin coils, and then that coil coils up, and then those coils coil up into what we call a supercoil. And the result of this is that this long stringy DNA gets compacted or condensed, is the technical term, into this chromosome shape that you are seeing here. Okay, so that's what happens to the DNA right before cell division, after replication occurs. And that's going to be important. Understanding how nucleosomes work and things like that is going to be important when we get into regulating genes a little later on. All right. Finally, there is another kind of nucleic acid that we need to talk about, and that nucleic acid is RNA. This is a chemical cousin of DNA. It too is made of nucleotides. This time, though, the sugar is called ribose and not deoxyribose, but it is a pentose sugar. It too has a 3' 5' end, just like the DNA. You number the carbons the same way. And instead of thymine, the base uracil is used. So that means anytime you would use thymine, instead you use uracil, and there's uracil. Okay, but notice that uracil is still a pyrimidine. It is composed of only 1 ring of carbons. Now, the other thing that's important to note here is that RNA is single-stranded. Now, it doesn't always appear to be single-stranded. If we look at our friend, the tRNA molecule here, notice the RNA actually kind of folds up into this, what scientists call a cloverleaf structure. And it looks in places like it's double-stranded because you do get some complementary base pairing going on here, but really this is one long molecule that's folded back on itself. There are other RNA molecules that take on other shapes as well. The 3 that you really need to know about are the messenger RNA, which usually stays in this long, unfolded, it sometimes when it's synthesized it's not, but for the most part it's in this long unfolded molecule. This is the tRNA molecule, and notice up here there's a spot for an amino acid to bind to the tRNA molecule, because this molecule is transferring amino acids to the ribosome, and then also there's ribosomal RNA, which kind of folds up into a round shape, and it's a structural component in the ribosome. All right, now the messenger RNA molecule, which is the RNA we usually think of, this is the one that carries DNA's message to the ribosome, only contains 1 gene, and it's single-stranded. So because it's much smaller and much skinnier, RNA is able to fit through the nuclear pores. DNA is a big molecule. And so you don't really use DNA. While DNA stores information long term, really the messages from DNA get written as RNA, and it's RNA that can actually leave the nucleus. Also, what's important to remember here is the genetic code, especially in a messenger RNA molecule, it's written in 3 base segments called codons. And in the next video, we'll get into why codons are important.